Hello, welcome back to the Turks and Caicos Reef Fund's Coral Observer Training course. My name is Alizee Zimmermann, and this is session number 3. In this session, we are going to be looking at diagnostic differentiation and what to look at when you're in the field observing reef health. So what are we looking at, and how can we 100% identify that we are indeed observing stony coral tissue loss disease? To help me with this section, I have employed Andy Bruckner's PowerPoint presentation on how to recognize and describe stony coral tissue loss disease lesions. This is one of the PowerPoints that I have been handing out to participants via email. So please, again, if you are interested in receiving these, just email me on Alizee, ALIZEE, @tcreef.org. And you will see that written on slides in both episode number 1 and episode number 4. So, Andy Bruckner is a research coordinator in Florida and works closely with National Marine Sanctuaries as well as NOAA, the National Oceanic and Atmospheric Administration. Move this over here for a moment. So the first thing is identifying and characterizing lesions in corals. As you swim along and start observing, you want to identify the presence or prevalence of tissue loss and any other associated unusual biotic or abiotic conditions. What we mean here is you want to see how many corals, if any, are experiencing tissue loss and what other conditions around could be affecting them. You want to then determine if these lesions are signs of disease, or are these signs of predation, overgrowth, competition, or physical damage. You then want to differentiate from other diseases once you've decided that, in fact, the lesion is a disease. And then you want to lastly say, okay, is it SCTLD? So we're going to go through these different steps over the next 40 minutes approximately. So field diagnosis. Step 1, identifying abnormal corals. So you're swimming along and you notice that these corals just don't seem right. Something's wrong with them. You're probably noticing large sections of white from afar. So what you're going to do is you're going to swim a little closer and you're going to say, okay, has there been recent tissue loss? Okay, by that I mean you can see denuded skeleton. Then you're going to think, okay, which species are being affected? You have a look around, see if there are more species. That links into your third question, which is how common is it? And also into the fourth question, which is this spreading? Do you seem to see, can you observe a pattern of spread from coral to coral? Another question you're going to want to ask is it killing coral colonies? We talked about this a little bit in section episode number 2, which is that SCTLD usually has entire colony mortality. So you want to look around and see, are you seeing any recently dead colonies, corals around? And then, are there any unusual environmental factors at the site? Can you identify anything? And can you immediately identify a cause? For example, if you see a bunch of scraped up or mashed up coral, and then you see an anchor in the sand, well, then you have an immediate cause to effect relation there. So let's start with the beginning. You're swimming along and you see white on a coral colony. Now you need to decide whether it's bleaching or whether it's something else. So as you get closer, you get real close and you have a look, and if there is tissue present, now this implies that you can see little polyps moving, not necessarily moving, but you can see some indication of polyps, and you can see tissue. There is flesh there. It is not a denuded skeleton. Well, in this case, you're looking at bleaching. Something, be it temperatures or other environmental stressors, have stressed this coral colony enough that it has parted ways with its zooxanthellae, potentially temporarily, and that's what causes bleaching. If, when you look at it and you see it is completely denuded, there is no tissue present, Well, now we have two options. Now we have to figure out, is it predation or is it disease? So now you're going to look closer and you're going to be looking for skeletal damage, the presence of predators, the rate of tissue loss, the pattern of tissue loss, and whether there is any sloughing of tissue. If you recall in episode 2, I showed a close-up of an MCAV, a Montastrea cavernosa, that had sloughing tissue where you could really just see that live tissue melting off of the skeleton. So first thing we want to look at, when did tissue loss occur? So here on this beautiful photo of a Copophilia natans, a CNAT, boulder brain coral, we have a section over here, a patch on the left, old mortality, probably 6 months to a year. You can see that there's thick turf algae growing there. There's some sedimentation. But if I zoom in, I can see that there is a little edge here. Something's happening just along that edge there. And then if I look over here, now here I'm seeing some more. I've got some recent mortality here where I can see this bright white denuded skeleton. And then we have what we call transitional mortality. So if you look here, the transitional mortality is 4 to 14 days, and that's when we start getting that yellowy green turf algal growth. As I mentioned in episode 2, recent mortality is 1 to 3 days. So anything you're seeing that is bright white, that section of coral has died within the last 3 days. While we're on this photo, I do want to make a little mention about these lovely little four-eyed butterfly fish down here. They are corallivores. They eat coral polyps, and something that we've all been observing throughout the different regions or islands, sorry, in the region that have this disease is that all these corallivores are opportunistically feeding on easy meals. Necrotic tissue is easier for them to access than a healthy coral. So I've been seeing these little guys just nomming on this disease margin and then swimming off to a perfectly healthy coral of the same or different species and just going right for that one. So it's just to show you all the different ways that these pathogens can be spreading throughout our reefs. So we've now determined that it is a disease, or it's a lesion, sorry, it is not bleaching. Okay? So now we need to determine what the cause of that lesion is. Sometimes it's pretty obvious. You may see this this type of sponge overgrowth, which causes bioerosion. This is Cedel Cleona dextria, I think. It's a type of Cleona. It's an encrusting sponge, and you can tell that it's a sponge. It's got these valves, and it's just working its way and slowly engulfing a coral colony. You can see that. You can immediately say, okay, that coral is not healthy, but you can determine the cause of that. Other examples are competition or overgrowth. So this picture on the top right is, and once again, I know the acronym, but not the full scientific name, VASB here. That is a mat-like tunicate creature. And then on your right, you have an orbicella. Now, you can replace those with any 2 coral species or other encrusting organisms, and they're going to be fighting for space on a reef. So as they grow next to each other, eventually they're going to have their little tentacles are going to come out and sting the other guy, and this guy is going to send his tentacles and sting that guy, and they're competing for space to grow, or they're being overgrown. So this here is an orbicella that will be being overgrown by this VASB here. And so if you were to see these little bits on the edge here of denuded skeleton, but no other obvious lesions, and there's clearly 2 organisms that are next to each other, then you can make the decision or make the assumption that this is from competition or overgrowth. Galls, tumors, tube dwellers here on the bottom left, again, those are more obvious. They make these calcareous nubbins or tubes, and you can, again, determine that something is causing this. You may not know what, but it's not a disease lesion, and it's definitely not SCTLD. In the middle here, abrasion, that can sometimes be a little confusing. You've got these white sections that are denuded, But if you were to look closely at that, you would see that the skeleton is scraped. It is not a perfectly intact skeleton. So the other thing to notice is that it's only on the top rather than inside the grooves, which means it's maybe someone's fin or something, and it's just the part that they can access. They can't get into the grooves. And then the last thing you're looking at, again, aggression. So again, this is if you've got, for example, here on the bottom right, a Christmas tree worm right next to an agaricea. Well, it's going to be coming out and just attacking and making space for itself. The numerous animals of the underwater world. Perhaps it's not a lesion, perhaps it's other causes of mortality. So we want to look around and think of what's causing partial or complete colony mortality when we're down there. Sediment damage is one. As our region knows oh so well, the aftermath of hurricanes often leaves thick sedimentation in the water. This can then settle, and if the tides and water movement don't brush it off the coral and the polyps themselves can't move it off, eventually this will lead to some colony mortality. We have algal abrasions. So again, algae will settle and colonize small little sections, and they can start overgrowing and creating more space for themselves. So that can happen. And you would see the algae encrusting that area. Again, it looks very, very different to a complete disease margin. And the last one I want to look at right here is the damselfish nest. So damselfish are very unafraid little creatures, and they will go and they'll clear a whole section of coral, and they'll either create their nest there or they'll have algal lawns, and they'll also come right out and nip at your fingers if you get too close. So what are other causes? Well, predation. Predation is often something that will be mistaken for a disease lesion. So let's have a look at what different types of predation we have. So when you're down there and you see a lesion, well, first you're going to say, okay, is there a predator present? Is there a loss of underlying skeleton? Remember I said with disease, you have a perfectly denuded skeleton. With predation, it's usually scratched at or scraped. So you want to look for that. And then of course, the pattern of tissue loss can often tell you where that tissue loss came from. So fireworms, Hermidus predation, Hermidice. So fireworms consume the branch tip projections or knobs. So often they're associated with acroprid species. And I've been noticing them quite a bit. They will also come along disease margin. So they're actually often associated with diseased corals. And I do have a working theory, although I will say I don't have data on it as yet. But just as layman's observations, I've been noticing a lot more fireworms than I had been noticing before, and in larger congregations. Again, feeding on the necrotic tissue along the disease margin. So I'm thinking, you know, is there some type of opportunistic multiplication in the species? Like humans and other species, when things are going well and food is abundant, there tends to be more breeding. So perhaps something to keep an eye out. But either way, they are associated with disease, and they are often found feeding on the necrotic tissue. But if you just see one little lesion, just keep your eyes out and look around, especially around acroprids, which are not susceptible to SCTLD. You want to be looking for fireworms. The next little guys, similarly to fireworms that are also associated with diseased corals or necrotic tissue, are snails. And these guys can be quite cryptic. They can be hard to see. Their shells are often algae covered, but you do want to look around for them. So on this photo here on the top left of the Eusomilia fastigata, EFAS, here you can see that there's 2, 4, 5 snails just nibbling away at this flower cup here. Pardon me. But if you didn't see those snails, if you were to look in closer, in and around the stalks of those flowers, that's where you would see your snails. So just because you don't automatically see them, do keep your eye out. They aggregate, so they're often 2 to 50 plus. A couple weeks ago I was in, or a few weeks ago at this point, I was in Grand Turk, and I saw a coral with, I couldn't tell you, at least 50 or more snails just all on top of it. But I do believe that they had come to an already diseased coral and were just opportunistically feeding on the necrotic tissue. One of the ways to make a decision as to whether it's a disease margin or just snail predation, first of all, is how big that margin is. And secondly is the shape of the injury. Snails create a characteristically scallop-shaped injury or trail across the colony. And you can see that kind of in this top right image here, and even a bit on this image on the bottom left here. Believe it or not, these little mounds I'm putting my mouse on right here, those are snails. So again, you really need to look closely for these guys. But as you'll see when we get into the SCTLD part of this presentation, there will be so many other factors that will lead you to decide as to whether what you're seeing is stony coral tissue loss disease or potentially something else. However, especially if anybody's watching this from a country that does not yet have the disease but your neighbors do, these are the things that you really want to be concentrating on, because you want to be able to start getting your baseline data and start monitoring certain areas looking for the key signs. Okay, damselfish predation. So I mentioned the damselfish with the algalons and the nests, and they create on brain corals what is referred to as ridge mortality, the central photo here. That at first glance looks like tissue loss margin, disease margin. But again, it's only on top of the ridge. It's not in the groove, in the valley. And as such, this was created by something who couldn't access, their mouth is too big or their face is too big, couldn't access the actual valley. So they just took the tissue from the ridges. Parrotfish, white spot biting. So there's two types of biting that parrotfish do. We have spot biting, which is what I see more often than not here in the Turks and Caicos. That's one or more fish that just scrape in completely irregular shapes around a coral colony. Usually the tissue will actually regenerate in a few weeks. It just depends on how deep the scrapes are, and it'll leave some coral colonies with scarring. And then you have what they call focused biting. Now this is, again, this is Andy's presentation. Focused biting is not something that I have observed quite as much here in the Turks and Caicos. I mean, if I had seen this coral, my first thought would be some form of disease. However, again, if I were to be able to get closer to this in the field, I would see the scraping of that coral rather than a perfect denuded skeleton. So again, very important to really look closely at the corals. So now we've decided, okay, it's not predation, it's not something else, it's disease. So now we need to evaluate disease signs. These are all signs that something is, that there is a disease affecting the colony you're looking at. So we have abnormal, darker or lighter coloration. So we have a disease called dark spots disease, or bleaching, which is not a disease, rather it's a response to environmental factors. However, some diseases will stress a colony out, causing them to bleach, especially focal bleaching. Two, we have the presence of a pigmented band, a thick band of disease, bacteria, whatever that pathogen is comprised of, is just building up and moving its way along the colony. We have recent tissue loss. That's the kind of thing we would see with the disease we are currently discussing. We have unusual patterns of growth anomalies, show you that there is something genetically happening in there. And then some corals will actually show many different symptoms, multiple signs So they could have discoloration as well as a thick band. Again, because the discoloration often happens as a stress response when the coral is unwell. So, talking about bleaching and abnormal coloration, different corals will manifest bleaching in different ways. Usually they'll turn a creamy yellow-white color. Sometimes they are bright white. For me, most of the experience I've had with bleaching corals, they tend to have a slightly creamish tone to them. And that I just equate to the fact that there is still living tissue on the coral skeleton itself. But there are certain corals like the Psitterastrias, the S-cids, this middle photo down here, that bleach different colors, lavender, periwinkle blue, sometimes pink. With the S-cids, it depends on how deep they are as to which color they will actually bleach to. So bleaching-associated mortality is a potential and something that if you're watching in episode 2, when I spoke about the Parades, the mustard hill coral and the finger coral, the two Parades species, I noticed towards the end of last year that there were colonies that had segments of tissue loss. But these did not look like disease margins like we see with the other species that are affected by SCTLD. Rather, these were just sections. So I associated those to bleaching-associated mortality. And you can see on this top photo on the left here, you don't have a thick disease or even a thin disease margin. You just have a section that eventually just didn't recover. And so it died off and then got colonized by algae, turf algae, pardon me. And here, this is an image of recent tissue loss from bleaching. Again, you wouldn't necessarily know that right away, but you could tell because there's bleaching across the rest of this colony. Perhaps there are no other colonies nearby with rapid tissue loss lesions, or perhaps it's a species, like what happened with myself with the Parades, that is not known to be susceptible. So before saying, oh, well, then it must be susceptible, which it might be, you first want to look at other options as to why the tissue loss is occurring. I'm now going to spend just a little time. I'm not going to go too into detail on any of these diseases really, but just look at some of the observational differences between them and SCTLD. So Caribbean yellow blotch disease. Firstly, it primarily affects Orbicellas, but it can occasionally, so those are our star corals, our lobed, our great, and our mountainous, as well as the boulder. Occasionally it says that it can be found on Pseudodiplorias, which are two of our brain corals, or Amontostrea cavernosa. But generally speaking, you're looking at a yellow, often circular band that takes on edges of the coral, works its way from the base in, or from a spot that was already damaged. And the lesion will grow outwards. So it'll bleach the coral and grow outwards, but it really does create quite a yellow tinge to its circular lesion. And more importantly, I want to show you, it moves slowly. So this is at a later stage. So Caribbean blotch disease has now erupted on different sections of this coral. And you can see the inner sections have now been colonized with turf algae and thick turf algae, which is showing you how slowly that disease progression is. So that would be my first indication that it's not SCTLD, because for the turf algae to get that thick means that those sections died off months before. Dark spots disease. So I mentioned this earlier. Dark spots disease is abnormal discoloration, and it causes dark spots on different species. It affects Psitterastrias, the S-cids, and it's not that infrequent that I would see dark spots disease on these Psitterastrias here in the Turks and Caicos. Again, falling into that 2 to 3% of endemic disease on a healthy reef. It also affects our Agaricias, which I've noticed here, some Orbicellas and Montostreas. But for sure, for myself, I think I've mostly observed it here on Pseudoastrias and Agariceas. And it causes these dark blotching. And with the SIDS, I am going to zoom in with my fingers real quick. With the SIDS, it can cause these like mucousy patches that are coming off and creates blotching. So that's one of the reasons why the Pseudoastrias are kind of hard to determine which disease specifically is affecting them. However, they are one of our most common species on the reef, and not including them in our surveys would give us a very skewed view of our reef composition. So that's another reason to keep them. So then we have pigmented material overlying the tissue or skeleton. There are two options for this. Black band disease, which actually if you shine a light on it is actually kind of red, but black band disease causes a thick growth in a kind of irregular pattern, and it's very identifiable. It's just a thick kind of moosey growth along a disease line working its way outwards on a coral. Different to that though is Caribbean cilia infection, this photo on the left, and later on we will see more photos that have it, because this is more like very thin black hairs that are growing on a diseased or already recently dead section of coral. And the reason I want to pay a little more attention to the Caribbean cilia infection is that it seems again to be opportunistic, and we are noticing it, or I have been observing it at least, on quite a few colonies that have SCTLD. Once that disease margin is moving, then Caribbean cilia infection will just come in and start anchoring and growing in that area. So now we have tissue loss. Now we know that what we're looking at is some kind of tissue loss disease. Well, there's one tissue loss disease that's incredibly similar to stony coral tissue loss disease, but they're not the same. And it's called white plague. We know they're not the same because although scientists have not been able to specifically identify the pathogen or pathogens in SCTLD, they have at least identified that they're not the same as the pathogen which we know for white plague. Aside from that though, they're incredibly similar. White plague affects over 28 species and tends to work its way across corals again in bands that look very, very remarkably similar to SCTLD. One thing that's different though is that with SCTLD, you'll more often than not have several lesions showing up on the same colony, and also the spread of this disease that we're currently facing seems to be faster and more voracious than that of white plague. So now let's look at SCTLD specifically. Stony coral tissue loss disease, we have 22 plus susceptible species. Again, we're looking at the 14 most common and most susceptible of those species. However, I encourage everyone to, if you don't already have one, get yourself a copy of the Reef Health book, the Coral book by Paul Human. My copy is currently sitting on the table outside. And the more species that you are familiar with and that you can identify when doing surveys, the more valuable the information that you are collecting will be. So as with everything, the more information we have, the better. So the specific characteristics of SCTLD, you're looking at a distinct pattern of spread among species. So that's one thing we can say, okay, we know that maize corals are amongst the fastest and hardest hit. Well, if you're somewhere where you're in a place that doesn't yet have confirmed cases, you want to be paying particular attention to the maize corals, for example, and then maybe the pillar corals, and so on and so forth. You also want to look for the presence of colonies with unusual bleaching or paling. We call it focal bleaching in sections as well. As I said, a lot of corals bleach, at least sectionally, in response to the stress of disease. Lesions occur in multiple locations on colonies, so that is a big identifying factor. And affected colonies have prominent areas of recently denuded skeleton. So again, as I keep zooming into all those different sections there to really show you that sharp denuded skeleton, that is how you're going to differentiate it with bleaching or other potential ailments as well. Once the disease is established in a region, there is high prevalence. And that is true. We can go along the reefs of Northwest Point, West Caicos, Grand Turk. There is a high prevalence, South Caicos, there's a high prevalence of this disease. And once it's established on a reef, you're gonna start seeing it encroach to that 66 to 100% of corals of that species affected. Remember when we looked in section 2 at background disease and SCTLD specifically. And of course, high rates of colony mortality in the early susceptible species especially. And this disease stays active throughout the year and for multiple years on individual reefs. I've mentioned that Florida is on year 6. The Turks and Caicos, we are going into our 2nd year now. And this is why the preservation of reproductive tissue is so incredibly important, because we are trying to save these genotypes so that at least sections of these colonies remain intact to then reproduce. Now, I am not going to go through all of these colonies again, because these are the ones that we went through in section number 2 when we looked at susceptible species and how to identify them. But this is just to show you that Andy Bruckner also does have these corals on his slide, which again, if you email me on alizee@tcreef.org, you can get a copy of this as well as some other informational slides that can help with self-study. So again, this is an elliptical star coral, and this just shows you that there's several lesions happening at the same time here that are slowly popping up. This coral within a few days will look very, very different, unfortunately. Okay, so this is again just going through all those different species. So this is something that you can use in your self-study. And the other PowerPoint that I'll be sending people by email is AGRA's identification slides for those 14 corals. So those are great slides for initially learning those 14 corals and what they look like. I'm just going to click through this here now, and this was the last photo here. So Looe Key Reef in July of 2019. In August of 2019, myself and a representative from the DCR went to Key West for a learning exchange with MPA Connect, and we did actually dive the reef at Looe Key when we first got an opportunity to apply the coral treatment base that they're using in Florida, which is what the Turks and Caicos Reef Fund are also using. It was an interesting dive, but it was also a very foreshadowing dive, and I really do hope that we have the opportunity and ability to halt the progression of this disease on as many coral colonies as we can, so that our reefs have a chance to replicate and reproduce. So this is going to be the end now of section number 3, where we looked at disease differentiation and how to determine whether what you're looking at is stony coral tissue loss disease or potentially something else, and also just to have certain questions in the back of your mind anytime you're doing surveys in the field. Please do tune in for section 4. Section 4 is going to be looking at monitoring, how we do the actual monitoring, what our sheets look like, what the database is going to look like, and then just a quick conclusion and wrap things up. So thanks again for joining the Turks and Caicos Reef Fund's Stony Coral Tissue Loss Disease Observer Training course. See you soon.